Hello student, welcome to Anu's classes. Let's start today's video with one of the very important chapter, that is biotechnology principle and processes. This chapter is very important from the examination point of view. So I am going to cover all the topic of this chapter serially according to your NCERT book. So before starting the chapter, first come to the term biotechnology. So what do you mean by biotechnology? So bio means what? Bio means living. And this technology means technique. So all techniques where the living organisms are used to obtain desirable product which are useful for human. It means all the techniques where living organisms are used to obtain desired product which are useful for human, and that process is called biotechnology. Bio means living, so living organisms are used in this technology. So now come to the chapter. So in the first para of your NCERT book, there are three definitions of biotechnology are given. According to the book, the first definition is all biotechnology. So according to this definition, techniques of using living organisms or enzymes for organisms to produce products and processes useful for humans. It means according to this old definition, in biotechnology process, the microorganisms and the enzymes which are obtained from microorganisms are used in the technique to produce the product which are useful for human. Next come to the next definition, that is today's biotechnology or modern biotechnology. So biological techniques used for the manipulation of genetic material and fusion of cells beyond normal breeding barriers. It means according to this second definition, means modern biotechnology definition, in this process means these techniques are used to modify the genome of organism or living organism. Genome means the genetic material, means the DNA and RNA, and fusion of cells beyond the normal breeding barrier. It includes in vitro fertilization leading to a test tube. In this process, within the test tube, a embryo is developed. Then next is gene therapy. Gene therapy means within the test tube, the defective gene is replaced by a healthy gene, and that gene is introduced in a host to get the desired product. Then synthesizing a gene, etc. So these processes are come under biotechnology. So it means you you can say that by using this technique, desired quality product we can obtain. Now comes the last and most important definition, that is the European Federation of Biotechnology, EFB. And it is the most important definition, and generally question asks from this definition. So what a question may come, what is the full form of EFB? Then you have to write European Federation of Biotechnology. So according to this definition, that the integration of natural science and organism, cells, parts thereof, molecular analogous for product and services. The integration means mixing of natural science and organism, cells, parts thereof. Parts thereof means their parts. Molecular analogous for the product and services. It means in this process, in this technique, the natural science and organisms are used, cells are used, parts thereof means their parts are used, molecular analogous for obtaining product, a desired product and services. So in the first para of your NCERT book, these three definitions are given, and out of these three definitions, this European Federation of Biotechnology, this definition is very important. Now come to the next point, that is principle of biotechnology. So the biotechnology process is mainly based on two principles. First one is genetic engineering, second one is bioprocess engineering or chemical engineering. So genetic engineering, techniques to alter the chemistry of genetic material, DNA and RNA, to introduce this into the host organism and thus change the phenotype of the host. It means in this technique, first the genome means genetic material means DNA and RNA of a microorganism is changed, means modified, and then it is introduced into the host so that we can get the desired product. Because when a modified gene is introduced into a new host, the phenotypic character of the host will be changed. The next principle is bioprocess engineering or chemical engineering. Maintenance of sterile condition in chemical process to enable growth of only desired microbe in large quantities for the manufacture of biotechnological products like antibiotic, vaccines, enzymes, etc. So it means in the situation where the genetic engineering means modification of genome means DNA and RNA is going on, in that situation the atmosphere should be contamination free, means that atmosphere should be sterile. And this chemical engineering means they keep the atmosphere contamination free, because if that is contaminated atmosphere will be there, then what happen with this desired gene, another gene also developed, and as a result we will not get the pure desired type of characteristic gene. That's why to get manufacture or to get only the desired quality of gene, the atmosphere should be contamination free, and that comes under chemical engineering. And it means without chemical engineering, genetic engineering is not possible. So for to go smoothly the biotechnology process, both genetic engineering and chemical engineering are equally important. Now come to the next topic, that is gene cloning. So gene cloning means to make multiple copies of gene, and for the gene cloning process we need some requirements. That requirements are bacterial cell containing plasmid. We need one bacterial cell which carry the plasmid. Second one is desired gene. This is the desired gene. Restriction endonuclease enzyme which will work as a molecular scissor. Ligase enzyme which will work as a molecular glue. And E. coli cell which is work as a host. So these are the requirements for the gene cloning process. So now come to the process. So for the gene cloning process, first we need a bacterial cell. This is the bacterial cell which has plasmid. This is the plasmid. So what is plasmid? Plasmid is a self-replicating extra chromosomal genetic material. Means this genetic material has the self-replicating capacity. Since in the plasmid ori is present, ORI is called ori. Ori means this is the small space where the polymerase enzyme bind, and as a result it get the self-replicating capacity. That's why since ori is present within the plasmid, that's why plasmid is a self-replicating extra chromosomal genetic material, and this is the bacterial own genetic material. Then from this bacterial cell we have to isolate the plasmid. Why we have to isolate the plasmid? Because this plasmid will work as a molecular vehicle. Molecular vehicle means what? Molecular vehicle means, suppose this is a gene, we choose this is a desired gene, so we need this gene. Then this is a host where we have to introduce this gene. But this gene cannot be directly introduced into the host, since within the host it will not be able to maintain itself. So for that it need a vehicle who can, suppose this is the vehicle, who can carry the gene and introduce in the host, and then help the gene to maintain itself within the host, and then from this host this gene is transported to its offspring, and this vehicle will help the gene to do that purpose. So for that introduce a desired gene to a host, always there need a molecular vehicle, and here the plasmid will work as a molecular vehicle. That's why the plasmid will be isolated from this bacterial cell. Then suppose this is a human cell. In this human cell, this is the chromosome where the genes are, gene of interest means desired gene is present. So first this DNA is isolated from the cell. So in the DNA, this red portion is gene of interest, means this portion of gene we need, means the desired gene is this portion, the red portion. Then from this DNA, it is cut into small pieces with the help of restriction endonuclease enzyme, which is known as molecular scissor, and it is separated into small piece, and this is called gene of interest. And this cut piece of the gene of interest has these two sticky end. At the same time, using the same molecular scissor, means using the same restriction endonuclease enzyme, the plasmid also cut, and it has also in two sticky end at the cut portion. Then this gene of interest mix with this plasmid, and as a result, then with the help of the DNA ligase, which worked as a molecular glue, these two portions are jointed and they will look like this. And this is now called recombinant vector. Recombinant means mixing of two different genes. Since here two different type of gene join and form a single unit, that's why it is called recombinant vector DNA. Now this recombinant vector is introduced into the host where it can maintain itself, and since plasmid has the self-replicating capacity, so it will multiply its and form many number of the same plasmid, and then during the cell division it will go to its offspring and lots of recombinant gene will be produced. So this is the gene cloning process, means in gene cloning process the multiple copies of desired gene is produced. The first artificial recombinant DNA molecule was first formed in 1972 by two scientists, Stanley Cohen and Herbert Boyer. And in this process, what they do, they took a cell of Salmonella typhi. Salmonella typhi means the organism which causes the typhoid. So first the plasmid from this Salmonella typhi is isolated, and at the same time here another gene of interest is also isolated. They choose the gene of interest which is antibiotic resistant gene, which has antibiotic resistant gene. Then this antibiotic resistant gene mixed with the Salmonella typhi, kept in a beaker with this, and then by the restriction enzyme, first this plasmid and this antibiotic resistant gene is cut into pieces, and then with the help of DNA ligase, means molecular gum, these two are jointed, and as a result new recombinant gene is produced, and which is when hosted into the cell of E. coli, then the antibiotic resistant E. coli cells are developed. So like this the first artificial recombinant DNA molecules was formed. So in this process question may come, what was the first they took the cell? They took the cell of Salmonella typhi, and what was the name of the host cell? The name of the host cell is E. coli. So till now what we have learned? We have learned the three basic definitions of biotechnology, old one, modern one, and the EFB. Then we learned about the two basic principles of biotechnology, that is the genetic engineering and the chemical engineering. Next we learned about the gene cloning and the whole process of gene cloning. And last we learned that how the first artificial recombinant DNA molecule was formed, and what was the first cell was taken, that is Salmonella typhi, and what was the host taken for the first recombinant DNA molecule is E. coli. Now I am going to give you some tips. The basic steps to modify a organism genetically. OR means organic organism. Just I am writing here in short form. So identification of DNA with desirable gene. So in the first step we have to identify a DNA who is carried a desirable gene. Then transformation of the DNA to the host. After the recombinant DNA is formed, means after the mixing of the desired gene with the vector, means molecular vehicle, means vector gene, then it is introduced into the host, and that process means introducing into the host, that process is called transformation process. Then maintenance of the introduced gene within the host and the transfer to the next generation. So these three are the basic steps. First we have to identify, then we have to transfer the desired gene into the host, and next, last point, the gene has to maintain itself within the host, and then it should transfer from one generation to another generation. Now come for the tools of recombinant DNA technology. First one is restriction enzyme. Restriction enzyme means it will work as a molecular scissor. Second one is polymerase enzyme. Polymerase enzyme means help in the replication. Third one is ligase enzyme. Ligase enzyme means it is the molecular glue. Then is the vector. Vector means molecular vehicle. Generally plasmid work as a molecular vehicle. And last one is host organism. So for the recombinant DNA technology, we need these 5 as a basic requirements. So student, in my next video, I will discuss about the different types of restriction enzymes. Since this chapter has many topics to understand, that's why I will cover the chapter in 3 videos. Thank you.